Very much for the nice introduction, and thank you for the organizers for the invitation to speak at this event, which I enjoy very much. So I'm going to tell you today about one of the latest story coming out of my lab. We historically are a lab that works very much in lung cancer. So I'm going to focus the presentation today on this disease. However, we have generalized some of our finding in other cancer type, and I will show you some of these data. And the talk that I'm giving today is about a project that is now about, I believe, to be published. So it's not out yet, but hopefully it will be soon. And it's about a particular kinase called RSK4 or RISK4. And I'm going to show you today that it regulates drug resistance and metastasis in lung cancer, which are the two main causes for us failing to cure patients. And I'm going to also show you that we found a drug that is used in the clinic currently and that actually can target this kinase. So first of all, what is RISK4? So RISK4 RSK stands for ribosomal S6 kinase, and RISK4 is a member of the P90 ribosomal S6 kinase. There are four such risk isoforms in human. They're called risk1 to risk4. And what's characterized them is, first of all, that their degree of homology is very, very high. So they have over 85% homology. And another particularity about these kinases is that they contain two separate kinase domain, an N-terminal kinase domain that is believed to be the one phosphorylating downstream substrates, and a C-terminal kinase domain, the function of which at the moment is limited to its ability to activate the N-terminal kinase domain. This dual kinase organization is very particular to that family. So how did we come up with RISK4 as a possible target of interest? So we did a series of RNAi screen. First, we did a screen in bladder cancer where we looked at kinases that, when silenced, would sensitize the cells to either Taxol or cisplatin, which are two drugs that are used in the treatment of this disease. And we found that 23 kinases seem to be regulating the sensitivity to both drugs in our T24 cell lines. We then validated these 23 in 3 additional bladder cancer cell lines, and we came with 13 targets out of 23 that did validate in multiple cell lines. We then cross-correlated these results with the results of previous siRNA screen that we had done in lung, breast, and colon cancer, again for resistance to Taxol and cisplatin, and also a siRNA screen that we did looking at cell migration and invasion in lung cancer. And out of this cross-correlation between all these various siRNA screen, only one target came out as being present in all of them, and that was RISK4. So if we migrate only to our lung cancer results, what do we see in the clinic? If we're looking at RISK4 expression, we first see that RISK4 is not expressed in normal lung. So it is expressed at the mRNA level, but protein for RISK4 is undetectable in 100% of normal lung tissues. However, if you're looking at lung cancer, and this is all lung cancer types, you can see that about 60% of tumors are actually positive for RISK4 protein expression versus 43% only that are negative. And if we now deconvolute these lung cancer types into adenocarcinoma, squamous, and small cell, we can see that all these subtypes do express some RISK4 protein, but adenocarcinoma has got the highest proportion of positive cases compared to other lung cancer types, with almost 70% of lung adenocarcinoma showing RISK4 positivity. And again, if we look at the intensity of the staining, so the level of expression of RISK4 in these different subtypes, Again, lung adenocarcinoma comes with a far higher risk for expression as compared to squamous cell carcinoma and small cell lung carcinoma. And this picture is equivalent to what we see as well if we look at mRNA levels. So it's not only at protein level, but it's already at mRNA level. Moreover, this preferential treatment for adenocarcinoma is again seen at the metastatic site. So if we compare for the same patient, the metastatic site versus the primary site of the disease, in adenocarcinoma, we can see that over 50% of metastasis express even higher level of risk 4 compared to the primary tumor. And if we look at squamous cell carcinoma, this proportion is much lower at 24%. So it looks like there is something particular about risk 4 expression and adenocarcinoma as a lung cancer subtype. And this difference is seen again when we're looking at the survival of patients. So if we compare adenocarcinoma patients with squamous cell carcinoma patients, we can see that higher expressors for risk 4 do much worse than lower expression for risk 4 for adenocarcinoma patients, and this is highly significant difference. However, when we move to squamous cell carcinoma, this difference disappears completely. So again, it looks like risk 4 is particularly important in adenocarcinoma as a subtypes of lung cancer. So why is it important? And I told you at the beginning of this talk that we showed that risk 4 regulated the sensitivity of tumor cells to drugs. And indeed, here we use Taxol and cisplatin. This is in A549 lung adenocarcinoma cell line. And please remember that although all the results that I'm going to show you today are going to be in A549, we've reproduced these results in a panel of non-small cell lung cancer cell lines. So we know that this is reproduced in many cells of that lung cancer type. And what we can see is that when we treat with the cells with Taxol in the presence of silencing for risk 4, we can see that we have a huge sensitization of the cancer cells to these drugs. And this is due to the fact that we promote caspase activation. So when we take out risk 4, the caspase activation is promoted at lower drug concentration as compared to untargeted cells. On the other hand, if instead of silencing risk 4, we overexpress risk 4, we can see that in the cell line that already expresses risk 4, increasing further the expression of risk 4 make the cell resistant to the drug as compared to cells transfected with empty vector. And as opposed to this sensitization with caspase that we've seen here, the reason for that is that we decrease upon overexpression of risk 4 caspase activation, as we can see here through caspase cleavage and the cleavage of downstream targets. So we're inhibiting apoptosis in the presence of risk 4 expression. So I'm not going to show you these results here today in the interest of time, but what we've shown is that this was linked to control of the expression of BCL2 and IEP family members, and especially CIP2, and we can show that this sensitization is directly dependent on CIP2 expression. So this was for sensitization to the drug, but I also told you something about a screen that we had done for cell migration and invasion, and that risk 4 had come out as a hit there. And these are results that you can see here. So here we're looking at the ability of cells to migrate. And when we silence risk 4, we can see that we have a drop in the ability of the cells to migrate. And this translates to an inability of the cells to invade as tested through a collagen matrix. So here we've got A549, we've got decreased cell invasion, and this is an other adenocarcinoma cell line. And again, we can see that the cell silenced for risk 4 failed to invade. We can correlate this with a phenotype that we see in our cells when we silence risk 4. So here you've got cells that are control cells, and you can see that they're nicely separated and this is associated with their ability to migrate around. But when we silence RISC4, we start seeing a clustering of the cells, increase in cell-cell adhesion with increased cortical actin. And this is due to the fact that we are reversing the epithelial mesenchymal transition in these cells and making the cells more epithelial-like. And this is shown here by looking at EMT markers. When we silence RISC4, we can see that we've got an increase in E-cadherin, a marker for epithelial cells, and a decrease in N-cadherin and vimentin, which are markers for mesenterial cells, mesenchymal cells. So what we're doing is we're basically making the cells more epithelial, aggregating into sheets and repressing their ability to migrate. So the control of EMT markers is well known to be controlled downstream of a transcription factor called NF-kappa B. And so we wondered whether NF-kappa B activity was controlled downstream of RISC4. And here we're looking at the active part of NF-kappa B, which is relay. And this is an activity assays. And what we can see is that when we silence RISC4, be it in lung cancer cells or in bladder cancer cells, we have a drop in the activity of the NF-kappa B pathway. So we went on and characterized molecularly what was going on, and I'm not going into the details of these results, but only summarize it in this graph. So what we found is that RISC4 phosphorylates P300, which is an acetyltransferase, on a site that inhibits the activity of this enzyme. So normally P300 acetylates relay to sequester it away from the nucleus and into the cytoplasm. So when RISC4 is present and P300 is inhibited, relay remains in the nucleus and transcribe target genes, including EMT markers. Now, when we take out RISC4, what happens now is that P300 is activated, relay gets acetylated, comes out of the nucleus into the cytoplasm, and we fail to transcribe these genes, including the EMT markers that I just showed you in the previous slide. So this was all nice, it's all in vitro, but does it translate into any change in vivo? And so in order to answer this question, the first approach that we took was to use CRISPR technology to knock out RISC4 out of cancer cell lines. And the first experiment that we did was subcutaneous injection of tumor cells, having a xenograph of the tumor grow underneath the skin. And then once the tumor was palpable, we treated or not with cisplatin. And as you can see in the control cells here in blue, treatment with cisplatin doesn't lead to any significant change in tumor volume. And this is quite characteristic of lung adenocarcinoma cells. They're generally quite resistant to cisplatin. However, once you take out RISC4, as in this CRISPR RISC4 cell, you can see that now in the presence of cisplatin, you do get a response of these tumors that is highly statistically significant. So this was for the part of sensitization to drug. What about the metastatic phenotype, the invasive phenotype that I showed you about? So what we did is we took these CRISPR cells or the control cells, and we injected it through the tail vein of mice. And what happens then is that the cells go into the bloodstream and go and colonize selectively the lung. We then, after 8 weeks, extract the lung, section, multi-section these lungs, and count the number of metastatic sites and the area colonized in the lung. And what I hope you will see very clearly here, the red bar being the median, is that once we knock out RISC4, the cells are far less able to colonize the lung as the control cells. And this is, again, highly statistically significant. So all these results put together suggest that RISC4 is a potential very good target for lung cancer. But what we discovered is that if we want to target RISC4, it needs to be extremely specific. And the reason for that is that if we look at all these experiments that I showed you before downstream of RISC1, we find exactly the opposite results. So if we silence RISC1, we have increased invasion of tumor cells. If we silence RISC1, we make the cells resistant to chemotherapy. And unfortunately, if we silence both RISC1 and RISC4 together, we end up with no change at all in sensitivity. So the increase of resistance or the increase in sensitivity are basically nullified. And this is a problem because all RISC inhibitors that do exist out there at present are pan-RISC inhibitor and therefore inhibit RISC4 as well as RISC1. So what we understood is that we needed to develop a drug that would be specific for RISC4. So we went on and we did a drug screen, and I won't go through the details of this, how this was organized, but basically we ended up with 12 compounds out of a drug screen that selectively inhibited RISC4, one of which was Moxifloxacin. So Fluxacins are quinolone antibiotics that have been used for a very long time in the clinic. And as you can see here is with increasing concentration of moxifloxacin, you have decreased activation of RISC4. And what we can show, and I'm not going to go through the pharmacological data today, is that moxifloxacin acts as an allosteric inhibitor. So it's not a competitive ATP competitive inhibitor, but it's an allosteric inhibitor, which is a reversible and selective for RISC4. So we then went on and screened high numbers of the quinolone antibiotic of that same family. And what we found is that about 50% of fluxacins were active in inhibiting RISC4, such as trofloxacin, for instance, but that 50% of fluxacins were actually not active, as you can see here for ciprofloxacin. So this told us a very important thing, and that was that their ability to inhibit RISC4 was independent of their ability to be used as antibiotic, as ciprofloxacin is indeed a very good antibiotic. So the antibiotic properties of this molecule is totally independent from their ability of inhibiting RISC4. Next slide, yes. So what happens if instead of using it on the purified kinase, we use it in cells? Well, we reproduce all the effects that we see when we downregulate RISC4 with RNA interference or CRISPR. So here we use moxifloxacin, which was the first compound that we discovered. And you can see that with the red dots here, that in the presence of moxifloxacin, you sensitize the cells to cisplatin. And this is even more true in the presence of trofloxacin that is active at a far lower concentration. And you can see that you get the same response with sensitization to cisplatin. On the other hand, if you take ciprofloxacin, remember, this is one of the fluxacin that was not inhibiting RISC4, you cannot see any such sensitization. So we can directly link the sensitization to the ability of these compounds to inhibit RISC4. So this is again because of potentiation of apoptosis and increase in caspase activation of the cells that are treated with trofloxacin or any of the active fluxacin. So if we look now in terms of cell migration, we can see that trofloxacin is able to inhibit the cell migration of lung cancer cells, as well as cell invasion. So we basically reproduce all the biological outputs that we had seen downstream of silencing of RISC4 with using these compounds. So again, these were in vitro experiments. So what happens in vivo? So we used again a xenograft model using A549 lung cancer cells. And when the tumor was palpable, we gave either cisplatin or trofloxacin alone or in combination. And as you can see here is in this waterfall plot, treatment with cisplatin, as we've seen before, is not good enough in terms of controlling disease progression, as there is very little difference between the control condition and treatment with cisplatin. But when you start combining trevofloxacin and cisplatin, this is the only condition where you have either a stop in tumor growth or even regression of tumors. And if we go into another model, that is that of a GEM model, so a genetically engineered mouse model for lung adenocarcinoma, you can see the difference here in the CT scan between the vehicle control or the combination treated animal, which show far less tumor burden. And I do not show you the picture for the cisplatin treatment because it is virtually indistinguishable from the one of the vehicle control. So next, we wanted to understand how trevofloxacin was working. So we first started by crystallizing the N-terminal kinase domain of RISC4, and you have the crystal structure here. Unfortunately, we were unable to co-crystallize RISC4 with trevofloxacin because the crystal then dissolved. So we had to resort to another strategy to detect binding of trevofloxacin to RISC4. And so we used deuterium exchange mass spectrometry, which showed an area of binding for trevofloxacin in the C-terminal domain of the N-terminal kinase domain. We then went on to characterize that part of the molecule. And the way we did this was to, sorry, I've got a problem going to my next slide for some reason. I'm sorry about this. Okay, and so what we did is we used Markov transient analysis, which is a mathematical approach to detect the presence of an allosteric site on our protein. As I told you, trevofloxacin is an allosteric inhibitor. And so we found that the region that we had determined by mass spectrometry to be the proposed binding site for trevofloxacin was indeed an allosteric site that was very connected to the active site of the protein, as shown by the probability distribution here in red. And our models also showed that while in the control condition, you have propagation of signal from the allosteric site to the active site through the activation loop, this propagation is blocked in the presence of trevofloxacin, meaning that trevofloxacin prevents the link between the allosteric site and the active site. And the reason why this takes place is because trevofloxacin bind exactly in the part of the N-terminal kinase domain where normally the hydrophobic motif, which is a part of the linker region between the two kinase domain of RISC4, binds. And this binding is a way for the protein to inhibit itself. So it's an auto-inhibitory approach, whereby before activation of the kinase, the protein is folded with the hydrophobic motif binding into this region. So basically what happens when we use trevofloxacin is we reproduce this natural auto-inhibitory approach and we prevent RISC4 activation. So in summary, what I showed you very quickly here is that RISC4 inhibition sensitizes lung cancer cells and also bladder cancer cells to chemotherapy and inhibits invasion in vitro, but also in vivo. And this is through a combination of inhibition of relay activity that controls the invasion and migration, but not the sensitivity to chemotherapy, and through controlling the production of anti-apoptotic molecules of the IAP and BCL2 family, which is now controlling the sensitivity to chemotherapy. So the RISC isoforms do not share many of their biological function, despite large sequence conservation. So RISC1 and RISC4 actually do have opposite biological function in terms of chemo sensitivity and invasion. And so we need to specifically and selectively target RISC4. And I showed you that some fluxacins are good chemical starting points to actually specifically inhibit RISC4. Trovafloxacin is at present our best compound in terms of activity, but levofloxacin also works quite well. And what is interesting about levofloxacin is that a clinical trial had been done many years ago where they looked at levofloxacin as an antibiotic to prevent infection following treatment of lung cancer patients with chemotherapy. And what they found is that patients treated with levofloxacin had an extended overall survival that was independent from the ability of this molecule to prevent infection. And I think here we found the reason for that, and that is because of the ability of this protein, this molecule also to inhibit RISC4. So I thank you very much for your attention. This is a recent picture of my team. Luxemon, Stelios, and Rajat have been very much involved in this project. And Michael Seckel runs the lab together with myself. And none of this research would have been made possible without our great collaborators that helped us with various aspects of this story. Thank you very much for your attention, and I'll take questions now.
Thank you, Dr. Oliver, for your amazing presentation about targeting RSK4 prevents both chemoresistant and metastatic in lung and bladder cancer, potential of repurpose fluxacin and as novel therapeutic agent. Thank you. If there is any question, you can ask Dr. E Pardo.
Yes, myself, Rameshri here again. Thanks, Dr. Oliver, for the very useful and informative presentation. Indeed, it added questions for my doubts. One thing is continuation with the old presentation was presented by Dr. I had asked ovarian cancer marker as a CA125. Taking one of the example as a case, what happens if one individual has an ovarian 20 years back and it turns to be as a lung after 20 years? So will you consider ovarian as the primary, or how do you stage that into the lung? Because basically it has a recurrence. So when you say it's a lung cancer now as a metastasis, so will you consider and give a treatment for a primary as an ovarian, or will take as a metastasis as a treatment and take the next level for a metastatic lung cancer?
So I think here, as far as we understand this, so we haven't looked at the expression of RISC4 in metastasis from other cancers in the lung to see, for instance, whether the lung is the environment that enables the expression of RISC4 in tumor. But we don't really think that this is the case. So we think that there is something particular about a particular cancer types and RISC4 expression. For instance, if we go to breast cancer, it seems that the results that I showed you today do not pan out. And there have been some reports saying that RISC4 is actually a tumor suppressor in the breast. We don't exactly understand why there is this difference between cancer types. It might be because RISC4 is not a single molecule. There are variants of RISC4, so there are isoforms of RISC4. And we think that there might be difference in which isoform is expressed in which cancer. And so depending on which isoform is expressed, it's either a tumor suppressor or an oncogene. So I think it's very important to look at the cancer specificity when you're assessing this. And in response more directly to your question, at present, with our knowledge, I would consider the metastasis of the ovarian tumor in the lung as an ovarian cancer. And I would not necessarily assume that targeting RISC4 would be beneficial in that case. Did I answer your question?
Yes, continuation with the answer that I received. As you said, the RISC4 will not be applicable as for this. So the option of the next would be targeted therapy, because I didn't see bevacizumab as a molecule mentioned in any of the presentation. Maybe I missed out, I'm not sure. So can the targeted therapy of bevacizumab will not be resistant to it? Like, could you just give an insight of it?
We don't actually know. So this is something that that we intend to look at in the future. We also think that RISC4 to some extent regulates the inflammatory phenotype and possibly the recruitment of immune cells. But this is something that for which we're looking for funding at the moment. So we haven't started this research yet. So I'm afraid I can't answer this question right now.
Okay, thank you.
No problem.
Hi, Professor Pardo.
Yes.
Yeah, I have a question. Thanks for your interesting presentation, and I'm interested in your animal experiment. I would like to ask you, did you what kind of vector did you use? Lentivirus or adenovirus? And did you do stable transfection or transient transfection in the animal experiment?
So you're talking about the CRISPR cells. So for the CRISPR cells for the animal experiment, these were stable CRISPRs, and we used a lentiviral vector. So these are cells that when they knock out, they stay knockout, and yeah, so they're stable.
Yeah, stable transfection.
Yeah. So for the metastasis assay, you can use transients because the basically the cells after 48 hours would be cleared out of the blood circulation anyway. But we consider that because of the effect of RISC4 on the growth of tumor as well as the metastasis, it would be a far better, cleaner experiment to do with stable cells.
Yeah. Thanks for your answer.
No problem.
Please, can I ask a question?
Yes, please.
I have raised my hand. Thank you, Professor Pardo, for your nice presentation. I would just like to find out your control in the animal experimentation, your control animal. How will you explain the reduction in the tumor size? Were they immunocompromised, or were they normal immunized, right?
So sorry, I didn't understand. In the control?
Yes, in your control group.
Yes.
If you show the slide on the
all right.
Control group.
Let me share. Control group. You mean in this experiment or?
Yeah, in the animal experimentation where you induce
these are independent mice. So this is the percent change in tumor volume. So you can see that you've got almost 100% increase in tumor volume. So this is not reduction. This is these are individual
okay.
mice. So you can see that in the control, all the mice have increase in tumor size, while in the combination, you start seeing either no increase or decrease in tumor size.
Okay. Thank you very much for the precision.
No problem.